Cancer stands as a complex disease in modern medicine, marked by uncontrolled cell growth and the ability to metastasize. Grasping its molecular mechanisms is vital for effective treatment. In this presentation, I will explore cancer biology using insights from biomedical foundations of medicine to show how molecular changes drive cancer's hallmarks. We'll cover 8 areas: cell signaling, genetics, cell growth, biochemistry, cell biology, immunology, pharmacology, and resistance mechanisms. Cancer starts when cellular signaling pathways break down, often due to faulty tumor suppressor genes, which are vital for keeping cells in check. For example, ARF binds MD-M2 and blocks it from destroying P53. This stabilizes P53, known as the guardian of the genome, so it can stop the cell cycle or trigger cell death if DNA damage occurs. BRCA2 repairs double-strand DNA breaks through homologous recombination to maintain genomic stability. SMAD4 works in TGF beta signaling to control gene expression and limit cell growth. E-cadherin keeps cells glued together to prevent metastatic spread. When these genes mutate, like TP53 in Li-Fraumeni syndrome or BRCA1 in familial breast cancer, signaling fails. Cancer cells exploit these weaknesses to grow uncontrollably. As signaling crumbles, genetic instability rises and drives cancer forward. Genomic instability marks cancer cells as fundamentally chaotic. Normal cells rely on robust DNA repair systems to preserve genetic integrity, but cancer cells rack up mutations and chromosomal chaos due to broken pathways. Take mismatch repair. Defects in genes like MSH2 or MLH1 disrupt error correction during DNA replication, sparking hereditary non-polyposis colorectal cancer. Homologous recombination can also fail, as seen with BRCA2 mutations, leaving DNA breaks unrepaired and genomes fractured. This instability lets cancer cells evolve fast, dodging pressures like chemotherapy or immune attacks. It also breeds tumor heterogeneity, where diverse cell populations within a tumor thwart treatment efforts. Each genetic misstep amplifies the next, creating a vicious cycle of disorder. With this foundation of genomic chaos established, cancer cells gain the capacity for the unchecked proliferation that drives their aggressive nature. Cancer cells override normal growth limits by hijacking nutrient-sensing pathways like mTORC1 and AMPK signaling. In healthy cells, nutrient scarcity or low energy, signaled by a high AMP to ATP ratio, activates AMPK. This shuts down mTORC1 and blocks anabolic processes to conserve resources. Cancer cells, however, evade these breaks through mutations, sustaining relentless proliferation even under stress. Meanwhile, cyclins and cyclin-dependent kinases, or CDKs, orchestrate cell cycle progression in normal cells. The F box protein FBW7 tags cyclin E for destruction, preventing excessive division. In cancer, mutations in FBW7 or CDK regulators disrupt this control, unleashing rampant cell cycle advancement. These molecular hijacks empower cancer cells to multiply unchecked, defying physiological constraints. With growth controls dismantled, cancer cells next rewire their biochemical machinery to fuel their rapid and unyielding expansion. Cancer cells survive by reprogramming their metabolism. The Warburg effect highlights their reliance on glycolysis over oxidative phosphorylation, even with oxygen present, channeling glucose into energy and building blocks for rapid proliferation. Glutamine further supports this, fueling the TCA cycle and nucleotide production to drive growth. Angiogenesis also sustains tumors by forming new blood vessels, orchestrated by pro-angiogenic factors like VEGF, which overcome inhibitors like thrombospodin 1 to deliver oxygen and nutrients. Additionally, cancer cells evade apoptosis, the programmed cell death process that removes damaged cells. Normally, mitochondrial stress releases cytochrome C, triggering caspases to break down the cell, but cancer cells block this, often through BCL2 overexpression, ensuring persistence under stress. These metabolic adaptations bolster tumor survival internally, while interactions with the surrounding microenvironment play an equally critical role externally. The tumor microenvironment shapes cancer progression and metastasis. Cellular senescence, a state that permanently arrests damaged cell division, serves as a natural shield against oncogenesis by halting proliferation. Cancer cells overcome this barrier through telomerase activation, which maintains telomeres, and by silencing tumor suppressors like p53, allowing relentless growth. Epithelial mesenchymal transition, or EMT, boosts motility and invasiveness, shifting cells from epithelial traits marked by E-cadherin to mesenchymal features like vimentin expression. This process drives metastasis, mirroring its role in wound and healing in embryonic development. But in cancer, it unleashes aggressive spread. Myeloid-derived suppressor cells, known as MDSCs, further aid tumors by crafting an immunosuppressive niche, shielding malignant cells from immune detection and attack. These microenvironmental factors advance cancer, revealing the immune system's dual role in both limiting and supporting tumor development. The immune system exerts a dual influence on cancer, curbing tumor growth while sometimes fostering it through chronic inflammation. Biomarkers of effect track early biological shifts between exposure and cancer onset, offering key checkpoints for risk evaluation. Chronic inflammatory states markedly elevate cancer risk. AIDS correlates with non-Hodgkin's lymphoma, asbestosis with lung cancer, gingivitis with oral cancer, and inflammatory bowel disease with colorectal cancer, revealing inflammation's pivotal role in oncogenesis. Type 1 interferons, vital for antiviral and anti-cancer responses, arise from intron-less genes on chromosome 9, with the IFN alpha subfamily comprising 14 variants. Dendritic cells drive cancer immunity as myeloid and plasmacytoid subtypes boost MHC2 expression after toll-like receptor activation, sharpening antigen presentation to T cells and bolstering anti-tumor defenses. Yet, despite immunological progress, drug resistance persists as a formidable barrier in oncology. Cancer pharmacology aims to craft therapies targeting specific facets of cancer biology. Most anti-cancer drugs block DNA synthesis, but L-asparaginase stands apart, halting protein synthesis by depleting asparagine, a nutrient critical for fast-dividing cancer cells. Drug efficiency hinges on IC50, the concentration needed to inhibit 50% of target activity, while selectivity, its reciprocal, reflects specificity for cancer cells versus healthy ones. These metrics guide the design of potent therapies with reduced toxicity, yet chemoresistance complicates treatment. Skin and colon stem cells resist drugs through enhanced DNA repair, elevated drug efflux pumps like P-glycoprotein, and amplified hedgehog and notch signaling. Drugs like goserelin target gonadotropin receptors, pentostatin blocks adenosine deaminase, and L-asparaginase starves cancer cells of asparagine. Overcoming resistance demands a deep grasp of its molecular roots, leading us to our final topic, cancer resistance mechanisms. Cancer drug resistance stands as a critical obstacle in oncology, reducing treatment efficiency and patient survival. Resistance can be intrinsic, with cancer cells naturally unresponsive, or acquired, emerging via genetic or epigenetic changes during therapy. Mechanisms include drug efflux pumps like P-glycoprotein expelling drugs, altered targets weakening binding, enhanced DNA repair countering damage, metabolic shifts creating survival pathways, and the tumor microenvironment shielding cells. For instance, vincristine disrupts microtubule polymerization and mitosis, yet resistance can blunt its impact. A new drug, cytarabine, inhibits cytosine deaminase, raising intracellular ERA DCTP levels in tumor cells, potentially heightening sensitivity to itself. Conversely, folate deficiency in patients on 5-fluorouracil impairs its ability to block dimethylate synthase, reducing deficiency. Cancer stem cells with robust self-renewal and repair amplify resistance, while increased tumor heterogeneity further elevates chemoresistance risk by diversifying cellular responses. Precision medicine uses genomic sequencing to detect resistance early. Combination therapies target multiple pathways, and immunotherapy bypasses traditional hurdles. Addressing resistance demands computational modeling, biomarkers, and innovative delivery, like cytarabine's approach, to improve outcomes. These insights, paired with today's cancer biology overview, push us toward more effective strategies. Grasping cancer's molecular mechanisms from disrupted signaling to genetic instability, altered metabolism, immune evasion, and drug resistance is vital for improving treatments. Each facet offers challenges and openings for therapy, By uniting insights across these areas, we can craft precise, personalized strategies to tackle cancer's complexity. As knowledge grows, so does our power to shift cancer from a deadly threat to a manageable condition, advancing patient outcomes.